So the topic of the presentation today is removal of cytostatic drugs from wastewater by an anaerobic osmotic membrane bioreactor, and it's an article published in Chemical Engineering Journal. So in this study, some of the cytostatic drugs from the wastewater were actually removed by the application of anaerobic osmotic membrane bioreactors. So actually the trace organic compounds including personal care products and some of the pharmaceutically active compounds and disinfection byproducts and industrial chemicals have gained concerns because of their uh, effect on environment and human health. So among the different trace organic compounds, the cytostatic drugs are also some of the compounds that are posing threat to the environment and that of the human health directly and indirectly. So cytostatic drugs are actually used as tumor treatment compounds, and they can enter into water cycle uh, from the hospital effluent like household wastewater, production discharge, and drug waste disposal. So they should be removed from the wastewater in order to pose any threat to the environment and ultimately to the human health. So wastewater treatment as control strategy was applied to remove these toxic compounds from the wastewater and from the water and to prevent the environment from danger. So the previous studies have demonstrated that the conventional wastewater treatment processes like activated sludge process were not able to achieve satisfactory removal of cytostatic drugs on the basis of biodegradation and adsorption to the biomass. So because of this limitations, membrane bioreactors have been applied as an alternate technology for removing cytostatic drugs. But the membrane bioreactor uh, technology and its removal efficiency has some limitation in order because of the uh, hydrophilic nature of the cytostatic drugs, as it can be seen that uh, some of the authors reported the 90% removal of hydrophobic anthracyclines due to the adsorption, while only a moderate elimination was achieved for hydrophilic cisplatin and carboplatin. So there is a limitation because the MBR cannot remove, or the lower removal of the hydrophilic substances and less biodegradable cytostatic drugs were not achieved through removal efficiency of 100% were not achieved through membrane bioreactors. So there were some limitations of these membrane bioreactors. So the recent advancement in the membrane technology have demonstrated that forward osmosis is an alternate or effective alternative to conventional membrane processes for seawater desalination and water reclamation. So the authors in this paper were inspired by the latest development of osmotic membrane bioreactors, and in which the forward osmosis membrane is used to retain dissolved organic matter in addition to suspended solid and biomembrane. So in the current study, an anaerobic osmotic membrane bioreactor for cytostatic drugs removal was applied, and why the forward osmosis membrane was used, so because of its high retention nature and the residence time of the trace organic compounds can theoretically approach the sludge retention time in an osmotic membrane bioreactor. So they have a so this reactor, anaerobic osmotic membrane bioreactor, is a good alternative for removal of these cytostatic drugs. So the experimental setup and operating conditions that were used in this paper can be seen in this slide. So as it can be seen that the membrane that was used was a flat sheet FO membrane made of thin film composite TFC polyamide, and the configuration of the its membrane is the along with the reactor is that of the submerged membrane reactor. So this submerged membrane reactor that it can be seen, the membrane is submerged in the system. So the synthetic domestic wastewater were applied to remove uh, the to remove all the means the compounds uh, to remove the compounds from that of the synthetic domestic water, and the total organic uh, total organic compounds, and that is ammonia, nitrogen, total phosphorus, synthetic wastewater were given are given in the slide, and their values. While the pH of the synthetic wastewater was around 7 and its conductivity was around 1.1 millisiemens per centimeter. So this is the setup that was used by the authors in their experimentation. So these are some of the cytostatic drugs tested in this study, which are the cyclophosphamide CPN, azathioprine, and methoxytrexate, and doxorubicin, and epirubicin, and flutamide, and methothane, and tamoxifen. So these are the cytostatic drugs that are studied in this study, and their removal efficiency by the membrane processes were analyzed. So the analysis of these cytostatic drugs, the sample preparation. First, they have prepared the sample by the sample extraction method, and the concentration of cytostatic drugs in the feed, water, sludge, supernatant, and effluent samples were determined by solid phase extraction. And for the sample extraction, they have used Strata XSP cartridges, and then the drugs were analyzed the mass by the mass spectrometry system, while the chemical oxygen demand and total nitrogen and total phosphorus concentration were determined by US EPS reactor digestion method, while the total organic carbon was determined using a total organic carbon analyzer, while the volatile fatty acids were analyzed by Perkin-Elmer HPLC system, and the particle size distribution of the sludge was analyzed by using laser scattering. So all these uh, analysis technique were applied to quantify all the related parameters of the current uh, experimental analytes. So coming to the results and discussion. So the first one is the water flux and salt accumulation in anaerobic atmospheric osmotic membrane bioreactor. As it can be seen that salinity the salinity as the salinity here increased, and from initial value of 1.2 millisiemens per centimeter to a final value of around 21 millisiemens per centimeter in cycle 1. So this salinity buildup can be attributed to the retention of solutes from the influent, and uh, and it can be seen that because of this salt buildup, the flux, the water flux or the rate of flux decline, as it can be seen from around from around 10 to that of the 2. So this decline this rate of flux decline is around 0.31 uh, liter per day in current study, was slightly faster than reported in the author's previous study. So because of so they have attributed this rate of flux decline to the biofouling of the membrane, and this biofouling of the membrane was induced by the polysaccharides, protein and microorganisms in the fouling of the TFC forward osmosis membrane, as was observed in the literature. But its permeability, the membrane permeability, can be effectively recovered by chemical cleaning, like using hydrogen peroxide like this. So one, and they have different cycles after the after first cycle, backwash and cleaning of, and this one is the, and then again the membrane was means the membrane was subjected to the wastewater, and so different cycles for 90 days period, 4 different cycles were carried out in order to check the effect of the salinity and the rate of decline of the water flux. So then this slide shows the removal of cytostatic drugs by the anaerobic osmotic membrane bioreactors. So as it can be seen that for all of the 8 cytostatic drugs studied in this compound, so all the means majority of the compounds were 100% removed, even at higher concentration of 100 microgram per liter and the lower concentration of 100 nanogram per liter. But for some of the compounds like azathioprine and that of the and azathioprine, so it can be seen that at lower concentration of lower dosage of 100 nanogram per liter, their efficiency was around like 95%. So this lower removal obtained at a dosing concentration of 100 nanogram per liter, it might be caused by the partial sorption of the compounds by the soluble organic compounds in the supernatant. And it can be seen apparently that the removal of the cyclophosphamide was 100% almost for a higher dose, that of 100 microgram per liter. So the cyclophosphamide was better removed, or effectively removed by this anaerobic osmotic membrane bioreactors, as compared to the conventional membrane bioreactors, where the efficiency was around 20% to 80%. And this higher removal of this compound by the forward osmosis membrane is because the small molecular weight cytostatic drugs can be retained by the forward osmosis membrane, whereas they leak through the porous ultrafiltration microfiltration membrane used in conventional membranes. So the prolonged exposure of the cytostatic compounds, or the retention time of the cytostatic compounds in the anaerobic membrane bioreactor, can further enhance the removal by the biomass through a combination of adsorption and biodegradation. So the this anaerobic membrane, this anaerobic uh, osmotic membrane bioreactors is efficient in the removal of cytostatic drugs. This slide shows the removal of the drugs by the uh, anaerobic sludge. As it can be seen in figure 3a, that the anaerobic sludge exhibit significantly different results on the removal of 8 cytostatic drugs. But regardless of the dosage level, more than 90% of the sum of the compounds like 90% like DOX, EP, and doxorubicin and that of the epirubicin, they are uh, more than 90% were removed by anaerobic sludge. But at the dosage level of 100 nanogram per liter, the removal efficiency of some of the compounds like methotrexate is around 60%, and for in case of the that of cyclophosphamide, it is around 32%. These different results on cytostatic drugs removal might be due to their different absorption and biodegradability in the anaerobic sludge, as stated in the literature. So, according to the different result on biodegradation and absorption, the cytostatic drugs were divided into several groups. The group 1, as it can be seen in the figure B, that the compounds that are some in the group 1 are those compounds which can be removed by the adsorption on anaerobic sludge, and the properties of these compounds are they are positively charged, and which may promote it to their adsorption to the negatively charged cell due to electrostatic interaction. The second group uh, can be attributed to the hydrophilic neutral compound like methox, like the methotrexate. Uh, it is mainly removed by the biodegradation. So it suggests that methoxin belongs to the easily degradable cytostatic drugs. And the group 3 compound contains those compounds which are the azathioprine and that of the like flutamide. They can be removed by the combination of adsorption and biodegradation, while cyclophosphamide can only be removed by that of the adsorption. This slide shows uh, the rejection of the drugs by the forward osmosis membrane, how the cytostatic drug can be removed by the forward osmosis membrane. So it can be seen in figure 4 that 8 cytostatic compounds removal by the forward osmosis membrane. So there is a like if we see in the slide, so the compounds that have the molecular weight 370 Dalton or greater than 370 Dalton, they are effectively removed by the forward osmosis membrane, maybe due to the size exclusion. But the neutral compounds with low molecular weight, like that of the molecular weight 277 Dalton, like this 277 Dalton, and uh, and 130 Dalton, they are not well rejected by the forward osmosis membrane. So based on the above result, it can be concluded that the rejection of the forward osmosis membrane was a dominant factor during the removal of the compounds by the anaerobic osmotic membrane bioreactor, while the anaerobic biomass played a more important role in the removals of the other cytostatic drugs. And this slide shows the effect of the cytostatic drugs on reactor performance. How, if the cytostatic drugs are present in the compound in the wastewater, so will they affect the performance of the reactor or not? So as it can be seen that in the that biogas is produced, so methane production, as it can be seen in the figure, like this. So the methane production, this is the methane production. So the methane production ranges from 0.18 to 0.23 liter per gram COD. So this is consistent with that of the previous studies by the author. So, but they have also said that this value dropped to 0.16 liter when the drugs was added. So this implies that inhibitory effects of the cytostatic drugs on the production of methane, and and the other this and the other this figure B shows the mean flock size. So as it can be seen that the cryostatic drugs not only disrupted the microbial metabolism but also affect the sludge properties. So when the cytostatic drugs were added, the mean flock size of sludge decreased from 92 from there from 92 to that of the 80 micron. So, so it has been reported that cytostatic drugs induce the increase of EPS concentration of sludge, especially protein and polysaccharide, as it can be shown. That is can be seen here that the introduction of the means when the drugs are introduced, so there is an increasing trend of the protein and polysaccharides. And it can be and according to the literature, it is caused by the stress response mechanism. But because of despite this detrimental effect of the cytostatic drugs, good quality of effluent was still maintained in the anaerobic membrane bioreactor. So in the conclusion, so in this study, the anaerobic membrane bioreactor exhibit excellent removal for all the cytostatic drugs, and this removal is owned by the anaerobic sludge, and that is the forward osmosis rejection. And the anaerobic biomass played a more important role in the removal of the compounds cytostatic drugs. And further analysis, they have showed that some of the cytostatic drugs were just removed through the adsorption, while some of the like some of the compounds, methoxythreate, can only be removed by the biodegradation. And some of the compounds are like flutamide and that of the methane, they are means removed by both biodegradation and adsorption. And it has also concluded that the cytostatic drugs adversely affect the methane yield, accompanying with increase of the volatile fatty acids, indicating an inhibition of cytostatic drugs to the microbial activity in the anaerobic membrane bioreactor. But despite the toxic effects on the microbial metabolism, the cytostatic drugs also influence the sludge properties, that reduction of particle size and increase of EPS. So that's all. Thank you.